All right, so now we're starting the recording. This is Bio 208. We did attendance, and I'll try to remember to do attendance before. Courtney, I did not see you on my 60A or 60B attendance roster. You might be on the 212 or 696 roster, so double check which section you're in, or it might be that you or you might be a late add and it isn't on there yet. Hold on, Courtney, let me check and see on attendance tracker if I have you, because I just downloaded this from Blackboard today. Courtney, do you know which section you're supposed to be in? And then I think I saw Lauren, you showed up, and I think I saw Xavier show up. Okay, let me switch back. I'm like on 2 computers and 3 screens. It's a little hot mess. Okay, 212. Okay. All right. So that's attendance. Okay. So I still have 1 person that didn't allegedly show up. So again, I think right now the 60A and 60B labs are completely full, or there might be 1 spot on Thursday. If this person doesn't show up and doesn't join, then again, there might be an additional spot open. I'm not supposed to let people in the lab beyond the 12. They're really strict on, they took our chairs out. Y'all are not supposed to work in groups. You're supposed to wear face masks. You're supposed to enter in the door one at a time, spread out. So if you are worried about lab for any reason, or let's say the numbers start ticking up and you're like, hey, we haven't officially gone virtual yet, but I'm nervous about coming to lab, just send me an email. It's not a big deal. I have 2 sections online, so it is very easy for me to just let you start coming virtually if need be. And if you have to take your test, again, I'm already having to put the test online so I can make it work that you will take your test. You will have to take tests with Respondus lockdown. If you have a Chromebook, I tried to put out there, or an Apple computer that doesn't let Respondus work, I tried to put out there how you can get loaner materials from the St. Louis library or the IT people. If you fail at getting equipment because they run out, our backup plan is take it in person. So come to campus and take it with me in person. Or worst case scenario, you're going to have to call. We're going to do a Zoom call on your phone, and I'll watch you, and you'll set your phone up to be able to watch you take the test on your computer, so I can make sure that you're not using your book like everybody else and any equipment. So the goal will be everybody takes the labs, and this is again on my list, in person if you're in these 60A and 60B sections. And if we all go virtual, we all go virtual. But if anything is done online, it has to be done with Respondus lockdown and monitoring. I tried to post in the Start Here area the tips about how to make sure you don't have any books or pictures or people around that could make the monitoring system flag you for cheating, and then I'd have to, look at your video and watch your whole video to see what's going on. And then again, if there is a continuing problem, I'll either have you come in person and take it in person, and you just won't get to use the lockdown thing, or we'll have to do the thing where you put your Zoom on and you let me watch you. Yay, I feel like Big Brother or a stalker there. And I'll do like Big Brother, and I'll watch you take your test. Okay, so again, 60A and 60B, and I'll get better at this. Because 212 and 696 sections are joining us, because this is their chance to also hear me live, it's going to be like, I have 40 people in the class right now. That can be upwards to close to 60. So that's why, remind me, or, I don't know if Blackboard Collaborate lets us yet. I feel like I should be able to take a snapshot of all the attendance somehow, but I haven't figured that out. But just email me if you worry like you showed up 10 or 15 minutes late. That you were there, send me a message. Okay, I think I got everybody again except that one name, Zahara Ashkar. That's the only person I don't know who's here or not here. Okay, Dora, can you turn on your mic. Dora is my former 208 student, and she is going to be your SI, which is now renamed as a PAL. So Dora, take over. Dora, we can't hear you. Can you chat in the chat or something? I don't know what's going on, Dora. Okay, she says 1 moment. I'm trying to make you a presenter, Dora. Okay, she keeps disconnecting or reconnecting. Okay. Dora is your SI or PAL. She should have a video link, and she should have something in the announcements. Hopefully everybody saw that.
Can you hear me now?
Yes, I can hear you now. All right, your turn. You get the floor.
So my name is Dora. I'm going to be your peer facilitator for the PAL program. And pretty much what the PAL program focuses on is helping students have a higher pass rate in classes that are generally harder than your average courses. So pretty much my goal is to help you pass this class and kind of enjoy it and retain some information for your future ventures in whatever direction you go in. I sent out a survey, and that survey just is to gauge what you guys are trying to get out of the class and what would be some good times for us to meet. We can meet either twice a week for about an hour and a half or 3 times a week for about an hour based off of whatever answers you guys give me in the feedback of the survey. I also provided my email address on the announcement that I made this morning, which is where you can also find the link for the survey. So feel free to ask me any questions or any concerns. But overall, I'm pretty much here to try and help this be a more productive, fun experience. And you guys have a really great professor. She does make it fun, and she will work with you. So I guess everyone's here just to see you guys succeed. And if you guys have any concerns or questions, reach out. I'll also make a video explaining what the PAL program is and how it kind of works, just so you guys can have a better idea. And that should be up a little bit later today.
Thank you, Dora. And I already gave Dora the topics that she should be covering for lots of Unit 1 information. Again, kind of helping her, and I've given her access to the ADI-LT. And since the ADI-LT labs are for completion, I am okay with you guys having Dora help you with them if that is what you want your sessions to be about once a week or something. You know, she can help go through the pictures or go through the things. Some of the information on there might be a little bit beyond what we're doing, but I thought it was good to do anyway. So she can be an asset for doing the online lab. She can be an asset for doing the study guides that I pulled and put together. Yes, I actually made study guides. I succumb to the whims of students. So there's a study guide available for you guys to look at each week. She can help kind of she doesn't answer that or doesn't go into that much detail on that. She can help kind of gauge how much maybe you need to freak out about points on a study guide and that, okay? So Dora will be, no, the Sally over at Academic Support said that Dora can only meet you guys online. She cannot do in-person stuff, sorry. All right, so our meeting times will be 11 to 11:50. Now, on the weeks we have test, I'm not going to have you guys meet on usually the Wednesday, so that way those of you that want to take your test on Wednesdays or tab on Wednesdays, you have kind of like some time allotted that week. You don't have to do a lecture and try to take your test. Usually the tests are going to be open. I'll get to your question in a minute, Nick. Usually, or Dora can answer it in the chat. Usually the tests are going to be open like after Mondays around noon. The test will open and they'll be closed by Thursday, 11:59 p.m. So you'll have about 72 to a little bit over that hours to try to take a test in a lab. Those of you that come to campus, you take lab at campus. If you want to take the test in person, you can either take it immediately after the lab, or you can come to the next lab, the next, like if you're a Tuesday lab, you'll come Thursday to take your test, or reverse it, or just take your test online, okay? That way, if you are kind of like, I have a Chromebook and I have crappy internet and I'm scheduled to come to campus, I just want to take both of them, or it's easier for me to just come to campus, you can take both of them at campus, okay? And those of you online only, again, it'll be the same thing. We'll usually have a session on that Monday to review the quiz or whatever activity we did the Sunday. Then the test will open up afterwards, and you can take the test, and then you can take the lab online. If something happens and you have to come to campus to take it, everybody needs to let me know and make that appointment so you can come in like right at 12:30 and start your test, and then I can monitor you until, you know, I have to go to lab, and then I can move you to take it there, okay? So that's kind of the plans, all right? I am anticipating that, like Thursday, we heard my kid's second grade teacher, her husband tested positive for COVID. So I'm going to assume that we're going to probably have something on campus that makes us all go virtual pretty quickly. So the joy of having two sections online is that I have to set everything up for them anyway. So in the event that we get the note, hey, we're all going online, virtual learning, we can quickly switch over and do that. That's also why those of you coming to in-person labs on Tuesdays and Thursdays, yes, you're in person 1 to 2:45, either Tuesday if you're 60A or Thursday if you're 60B. If, again, you ever feel uncomfortable, you ever get nervous, just send me a message saying, I can't make it to lab. COVID's making me nervous, or I'm not making it to lab because, you know, the mask, people wearing the mask on campus and I just don't feel comfortable, blah, 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 whatever it is. It doesn't matter to me. If you just say, I'm not comfortable coming, I can quickly and easily make it work that you can be virtual, because there's probably somebody in my virtual class that would really, really like to come to lab in person, and I'll just let them take your spot, okay? So that's kind of how this goes. All right. Any questions to Dora or any other questions to me about procedures? All right. You do have to fill out some type of app or online form, and I tried to make that form available, and I'm supposed to see the form that says you have not come into contact with someone with COVID, you aren't experiencing fever or symptoms, you know those standard questions they typically ask you. You're supposed to fill that out before every visit to campus, okay? Okay, are the quizzes and exams timed? Yes, everything will be timed. There's usually enough time that you should be able to look through it all, and everything is done to where you can see all the questions at once, so you can kind of skip around. Typically the quizzes, except for week one. So week one's quiz is going to be our respondus time to kind of test out respondus. But typically quizzes are going to be open book, open note, except for week one. And tests are closed book, closed note through respondus. They are going to be almost the exact same test, not exactly the same, but almost the exact same test that everybody would take if they took the in-person test. And the in-person test is only allotted about 55 minutes, so most of your tests will take you about 55 minutes. Those of you that have access office letters, you will need to email me those letters so I can set up online that you can get your extra time. If you are going to be using Testing Center for Access, you will need to set that up with me 24 hours prior, because that means you're probably going to campus and I'll have to get a test printed and sent over there or whatnot. So just make sure you're emailing me your letter and letting me know what it says so I can either set it up online to have that extra time, or we set up for you to take your things at the access office. Okay, 212 folks. Yes, welcome Grace, and whoever else is here from section 212 and 696, you are welcome. I'm not going to set aside more time to try to lecture again. So this is your opportunity, if you want to join live lectures, to come to live lectures. The downside is you'll have to probably wait 24 hours for this recording to show up on YouTube. And that is part of the reason why I'm going to start the recording after I take attendance to try to minimize how many names I call out. And I'm sorry if you are offended if I call your name. I do apologize. And I'm sorry if I slaughter your name. Just to be easy, Grenier is a little bit of a mouthful and it's French. Just call me Dr. G. How long before the exams or quizzes? Almost everything should be available except exams or quizzes. They'll usually be available the week. Like I said, the tests are going to be limited to, we'll have a session, typically a test week. You'll have a test starting Monday afternoon. It'll open up and be made available. And the test will be until Wednesday night, I think. And then the lab will open and you'll have until Thursday night to take it. The goal being those coming to campus are going to take the test Tuesdays and Thursdays. So the window of time for everybody to take everything is between Monday and Thursday, 11:59. So there should be plenty of time with your work and schedule to maybe try to figure out a time to take a test on Tuesday night, a test on Wednesday night, or a test on Tuesday night and a test on Thursday night. Okay. Quizzes are usually going to be open, like quiz 1 should be open now, and you have until Sunday, 11:59 to do them. All the study guides should be available if you go to the week or of the that we're doing it. All those study guides should be available. And then my old school objective for before I made weekly study guides, and the weekly study guides are just me grabbing the objectives from the Human Anatomy Society's page. So it's not that much different from the objective sheets I provided previously. Okay. All right. So are there any other questions? I think I covered everything that's on here. Okay. So chapter 1 is about, or the chapter 19, I should say, our first year. And those of you who do not have the Martini, Nath, and Bartholomew book, if you're using McKinley or Saladin or something because you took it at Flow Valley or Forest Park, you might have a different chapter. But blood is the topic, okay? And so the big thing in lab we need to do is we need to try to figure out what is blood. So what is blood made up of? What are all the little components of it? And so when you take blood out of somebody's arm, and some of you have had this done probably recently, you get this vial of blood. And so the point is that that is whole blood. And whole blood is something you can't see through. It is slightly more viscous or thicker than water, but it's not quite like Jell-O. All right. It's slightly, if you did a pH test on it, it's going to be slightly basic. So there's less free hydrogens in blood than there is in water. All right. And for men, if I was to take all the blood out of your body, it should be somewhere around 5 to 6 liters of blood, and women, you should have 4 to 7 liters. So men tend to be a little bit higher because the average male is typically like 5'8", I think, in the United States or in the world, and then the average female is about 5, I think 5'5" or 5'6". So just size-wise, men should have a little bit more blood because they're on average a little bit taller. Now, if you put a 5'8" female and a 5'8" male together, chances are they'd probably be about similar in the amount of blood you can suck out of them. It's about 7% of their body weight. But there still will be some differences. We'll learn about the number of red blood cells should be a little bit higher in a male just because of testosterone's influence versus the female. Okay? So, whole blood is, remember from chapter 4, it's a connective tissue, and it's a special connective tissue because the matrix is a non-living background, and the matrix in this case is mostly water. We call the matrix the plasma. All right, and that's the background non-living component of blood, the plasma. The plasma is mostly water, and so if I could blow this up, it would tell us that the plasma is somewhere on average for most people 60 to 50% of your blood. It's a little over half, and it's mostly water. Suspended in the plasma is going to be some protein, and some of the proteins we think of commonly now are antibodies. So when they do that antibody test for COVID, They could take your spit, your saliva, which comes from, again, leaking plasma that forms saliva. They could take your blood, and they could do a test of, do you have the protein in your blood that is made by your body to attack and fight the COVID virus? All right. Other proteins in your blood might be your albumin, which helps you carry protein or carry fat in your blood. Albumin also is an important protein for keeping the blood water content inside the circulatory system so it doesn't all leak out. And you also have some other carrier proteins. So you'll have some lipo carrier proteins like LDL, VDL, and those are the proteins we take in a cholesterol test. How many of these carrier proteins do you have to carry fat in your blood? And usually when you have a lot of them, you're carrying a lot of fat in your blood, and that's considered bad for heart health. Okay. Other protein carriers would be things like thyroid binding protein. You have testosterone binding protein. You have some metallic binding proteins like a ferrous or something that carries an iron molecule. So there's lots of proteins in your blood. All right, so it's the most numerous non-living floating suspended item in your blood, in your plasma. Okay. The other things you carry in your blood are some of your electrolytes, all right? And so these are some of your non-organic molecules, and sodium, potassium, chloride, bicarbonate are some of those molecules. You'll also have some organic molecules like glucose. You'll have small organic molecules like glycerine. You'll have creatine. You'll have drugs that can be in there. So some of your small molecules that are serving as something you took to help treat your headache or treat and medicate you. You'll have hormones. All right. So that's what's made of the non-living background material. All right. To kind of draw some similarities back to chapter 4, there is no collagen, elastin, and no reticular fibers in your blood. So that's another reason why it's a specialized connective tissue. There's no collagen. Collagen is going to be in the background connective tissue of the vessel and the heart. And so one of the things we should never have in our blood is collagen, okay, and elastin, proteins. And if they are there, that means we have a break or tear in the blood vessel, and you should have a clotting effect happen. All right. Let's see. The other part of a connective tissue is the living side. And remember, the living cells in blood are going to be majority red blood cells, white blood cells, and platelets. Now, technicalities, red blood cells do not have a nucleus. They have gotten rid of all their mitochondria, all their organelles. So technically, if you want to get into some of the, really finite details, blood cells are like on death row. They are terminally differentiated dying cells. They should live for about 100 to 120 days. They cannot replicate. They cannot respond to their environment. So they're kind of cells that are about to die, and we're using them as they're circulating around on this death row positioning to help us do some things, and then we get rid of them. Right? Platelets are also somewhat dead cells because they are also no longer truly cells, and they are going to not have a nucleus, but they do still have some organelles, some vesicles, and they are also about on a 2-week life ratio. So they are going to be pulled out of circulation if we don't use them in about 2 weeks. So of the living components of blood, 2 out of the 3 of them are kind of dying or dead or not technically still meeting the definition of a living cell. But they came from living cells, so that's why they fall into that living component. So we're going to call them the formed elements. White blood cells, they are going to come from the same stem cell that red blood cells and platelets come from, and many of them are trying to get to your tonsils, to your lymph nodes, to your spleen, to your liver, to your connective tissues. And so many of them are in the blood only for a short period of their life cycle. They may be hitching a ride just to get from, again, the bone marrow that they came from, the thymus gland they came from, then to their location they're trying to go and be part of the immune system. A few of them will stay in the blood system for longer periods of time. And what you're going to be asked to do, because they are in the blood, is you're going to be asked in this lab to start learning how to identify those white blood cells by the very big picture, gross macroscopic view in a microscope. All right. So red blood cells, if I was to look at the formed elements and I had 100 cells that I took out of your blood, 99.9% of the blood cells should be red. So that means out of those 100 cells I grab, almost all the time, all 100 are going to be red blood cells. And then every now and then, one of them might be either a white blood cell, a platelet. All right. So that's how numerous your red blood cells are. It's part of the reason why your blood is red in color. Now, the red blood cells have an iron ion embedded into the proteins that make up the majority of their intracellular contents. And iron, when it binds to oxygen, takes on either a more red, bright color when it's fully loaded up, or a less red color. Okay, so arterial oxygen-rich blood of our systemic circulation is going to be very, very bright red. And then venous blood is actually not blue. It's only look blue for veins when we look at it through our skin because of the way the light is being bounced off the fat in the skin and the coloring of the fat in the skin cells. But if you ever look at venous blood being drawn, it's pretty dark in color, and it's just because the oxygen attached is not as rich to the iron. Okay? So that's some of what's going on with whole blood. Now, when you get blood out of a person, if you put the blood in a container that prevents clotting and you spin the blood container, it is going to give you a separation of plasma and formed elements, all right? And you prevented clotting, so platelets are going to be formed elements. If you look at that division of about where does the water versus the cell components land when you spin things based on weight, that's centrifuging, all right? Again, your plasma should be somewhere around 55 to 60%, and your formed elements are going to be somewhere around that 45 to, I don't do math really well, 60, 40, 55, 45, okay? That number where we kind of draw the line of where the plasma ends and the formed elements begins, the percentage point is known as your hematocrit, right? And if anyone has ever been told they're anemic, chances are that line showed up around the point where your formed elements was 37% of your blood or less. And so what that is telling us is your ability in many cases to make red blood cells rich in iron, to make carrying capacity for oxygen is really, really low. What we would like to have available to our cells for normal cell function. And we call that anemia. And when you get into your nursing classes, and many of you want to be nurses, there's lots of forms of anemia, all right? And so we're not going to go into all the forms. Just know, in general, one of the big forms of anemia is when the hematocrit's below 37%, and it's because you probably are not making enough red blood cells rich with hematocrit, and that could be for a variety of reasons, and you're not carrying enough oxygen to your cells. Okay, so hematocrit would be if I took this vial, spin it, and I don't know if I can draw, and let's say the line shows up right there and it's maybe 50-50. So my hematocrit is going to be at the 50% point. So 50% of my blood is formed elements, 50% of my blood is plasma. Okay, sometimes when you get your blood drawn, they take like 2 or 3 vials. If you take blood and you let the platelets clot, when platelets clot, they are going to grab some of the calcium, some of the proteins, the fibrin, the fibrinogen, that typically suspended in the plasma. So if you let your blood clot and you spin it, you're going to get, again, a separation, but that separation is not true hematocrit because that separation is going to give you what we call serum. And serum, I'm trying to spell it, serum, which is going to have no calcium ions and no fibrinogen, which is and other clotting factors, because all of those molecules are going to be part of a clot that's going to form down here, okay? And so serum is in by definition, is a little bit more filtered plasma. So it's plasma that has lost its calcium and lost all of its clotting factors and fibrinogen. And so when we do certain tests, let's say for your hormone FSH or your hormone for seeing if you're pregnant, or for a really obscure antibody in your system, so maybe the COVID test, all right, you would want to take serum to try to find some of the more low numbered molecules versus plasma, because if you are taking plasma, there's more stuff in the water. If you're taking serum, you've kind of made some of the stuff in the suspension of plasma go away. And so it's a more filtered form of plasma. Okay, so if and ever you do blood work and you need to find something like maybe LH or FSH or the pregnancy hormones, they might actually ask that when they draw blood, they draw a blood into a vial that lets it clot, so they get serum instead of plasma, so they can do the test for that protein or that molecule with maybe less chance that it'll accidentally bind to a clotting factor that might be very close in origin or close in makeup to the antibody or the protein or whatever you're looking for. All right, so just be aware of that. Okay, so moving on to the red blood cells. All right, so review what are all the components and numbers associated with them in normal folks for all of your plasma elements and plasma components. All right. And then now we're moving into the formed elements, and we're going to talk about red blood cells. All right. We'll talk about white blood cells again later on in chapter 22, but this is our only chance to see red blood cells. So we need to spend a little bit of time on them. All right, so red blood cells are the most abundant of all of your formed elements. Again, they're 99.9% of the red blood cells. So again, if I take a sample of 100 cells, chances are all of them are red blood cells. If I do that 10 times, 9 out of the 10 groups of 100 are going to be all red cells, and one of them will have 99 cells of red and one of something else. All right, so in order to pack that many red blood cells, and there are millions of them in a droplet. So if you take a teardrop worth of blood, there's going to be millions of red blood cells in there. I need to make red blood cells so that they can be compactable, easily crammed into a very small volume. I need to make them to where they can be small and crammable, but still have a lot of hemoglobin binding protein capability. So the red blood cell is going to be a cell that is going to have a shape that lets it try to avoid getting snagged and broken and torn apart as it circulates in the blood. It's got to have a shape that lets it have what's known as a low surface to volume ratio, meaning that anywhere you sit inside the red blood cell, you're close to the cell membrane, so you can easily grab nutrients that diffuse in, and you can easily get rid of waste products that you accumulate inside the middle of the cell. All right, you need to have a stackable, so that way you can again cram a bunch of them into a small space. And so what the red blood cell has is this biconcave shape, and you can kind of see it right here. So it looks like a donut where the center of the donut is smushed in and it doesn't quite form a hole. All right, it's very round. And again, the rounding allows it like plates to stack. And that kind of structure it creates when you look at the stacking of them in a capillary, like right here, is known as roulette or rouleau, depending on if you're French or not. All right, so the red blood cell has a very unique structure in that it's biconcave, so it's very stackable. It has this amount of cytoplasm inside that whether I sit here or I sit here or I sit here, I am always close to the edge of the cell. So as something like oxygen comes in, I can really quickly, no matter where I am, grab that oxygen and bind it to my large number of hemoglobin proteins. Okay. The red blood cell does not have any nucleus, any organelles for the most part. It has gotten rid of all of those big features that normally exist inside the cell in order to make more room for more hemoglobin. Now, inside the cell, the cytoplasm still has some water, so it's still mostly water. It still has some sodium, potassium, some hydrogen, some bicarb. It still has some other, I don't mean to keep making all these lines, all right? It still has all these other proteins in here, and some of those proteins we're going to learn important ones like carbonic anhydrase that helps carbon dioxide and water in the cytoplasm convert into bicarbonate, and carbon dioxide and water convert to bicarbonate and a hydrogen ion. All right. There's going to be embedded in the protein some proteins that are going to be for letting a chloride ion come in and a hydrogen ion leave. There are going to be some proteins in here related to breaking down glucose to pyruvate. So the proteins of glycolysis. There is going to be some glucose inside there. So the red blood cell cytoplasm is mostly hemoglobin, but there are some other things in there. It's not completely just hemoglobin. But every time it got rid of a mitochondria, it got rid of a vesicle, it got rid of the nucleus, it got rid of ribosomes, that space that was created by getting rid of that organelle, it made and put in that space more hemoglobin protein. All right, and by having a large number of hemoglobin proteins, the hemoglobin protein has those 4 spots. Sorry, what am I doing? Okay, it has those 4, again, heme molecules at the center. All right, that's where the iron is. So this hemoglobin can bind an oxygen here, an oxygen here, an oxygen here, and an oxygen here. Okay. All right. Now, there's no cell nucleus. So if this red blood cell bumps into something and something tries to, let's say, take over it, this cell has no ability to try to ramp up and generate a response to that. It has no ability to respond to stimuli. So because of that, the red blood cell is on that limited lifespan of about 120 days. Okay, so again, all of this is in your chapter. All of this has been done in a previous lecture. I'm just trying to give you the cliff notes here because I only have about 8 minutes. All right, and here we go through what is the red blood cell life cycle. All right, so the red blood cell is going to be made in bone marrow. Red bone marrow is where it's made, and part of the reason why it's red is because blood flows through here, and the blood, as it flows through here, through capillaries, is going to allow all the signaling molecules to interact with the stem cells, the hematopoietic stem cells, to influence them to say, hey, start making more red blood cells, all right, or make other cells. And because that red blood from the circulatory system is flowing in here in higher amounts, it'll give it that red color. In addition, iron is going to be sent to this area. So as we need to make more red blood cells, iron is available to then bind up and put in those hemoglobin proteins. All right. So when a red blood cell leaves the bone marrow, it's going to leave, and you can't quite see it, but there are some little pieces of ribosome and message RNA still making probably human molecules in this cell, and it hasn't fully collapsed yet into that biconcave shape. And so it is a reticulocyte. And if I draw blood from you today, every one of us should have about almost 1% to 0.8% of our red blood cells in our system should be reticulocytes. So every day you take out 1% of your red blood cells. So if you have a million red blood cells, you're taking out 1%. So if you're taking out 1%, you should be adding 1%. All right, so every day there's about 1% of your red blood cells being added and about 1% of your red blood cells being taken away. All right, it takes about 24 to 48 hours for that reticulocyte to then convert into a mature red blood cell, and then that mature red blood cell flows through your circulatory system. Now, we can't avoid it. There's going to be times and places in our red blood cell flow that there's going to be high pressure, and there's going to be snags and beating up of the red blood cell. So there is about 10% of your red blood cells that over the course of their lifetime are going to explode, are going to no longer be intact, and they're going to what's known as lyse. And when they lyse, all of their insides spill into the outside of the cell. And so that is how some of our blood might have hemoglobin in the plasma where it's not supposed to be. All right, and might have, again, a little bit extra of the proteins that are inside the red blood cells located in the plasma of your blood. And that is how hemoglobin can sometimes end up in your urine. So if you have red blood cells exploding at a higher than normal rate, causing hemoglobin to be higher than normal in your plasma, and then being filtered into your urine. And once hemoglobin's filtered in your urine, you can't get it back. And so when you're peeing, you have a lot of hemoglobin in your PP. We could then trace back that there might be something going on with your red blood cells and their ability to not spontaneously break apart, or the integrity of your plasma membrane is being damaged somehow. Okay? Most of your red blood cells should not go through this, but go through a process of circulating for 120 days, and in the course of that, they somehow get seen as old. Don't ask me how. You can get your PhD on figuring out how we know a red blood cell is old. All right? And the red blood cell, when it's determined to be old, and when it's either in the liver or the spleen, and mostly in the spleen, but it can happen in the liver, it's going to run into these cells that live in the spleen that are known as macrophages. This is one of your white blood cell types that are known as monocytes when they're circulating in your blood. They're known as macrophages, and sometimes dendritic cells, sometimes pneumocytes type 1. So they get some different names depending on where they end up, their final tissue location. But in our spleen and our liver, they are just known as macrophages. Right? And these macrophages, when they are living in the spleen and the liver, they run into all the blood that through the circulatory system flows through there every beat, and they bump into red blood cells, and they're looking for red blood cells that are old, and they're also looking for any cell, red blood cells and anything else, that is beginning to express non-self antigens. And we'll get more into that on blood typing. All right. And when they find cells that are old, like red blood cells, they will phagocytose, break them down. They are going to try to recoup as much as they can from the plasma membrane. So they're going to try to recuperate the lipids, the cholesterol, the proteins, and break them back down to amino acids. And they're going to try to recycle all that into new red blood cells and new cells all throughout the body. So they're going to try to keep the iron, and they're going to try to help bind the iron to its binding protein and send all that nutrient, potential molecules back to the bone marrow and other cells. Okay. What ends up happening if you go back to hemoglobin, all right, this heme molecule with iron in its core, we can make it, but when we pull the iron out, what we get left with is a molecule that we can no longer do anything with. It's not a useful molecule that we can recycle into a new hemoglobin molecule. So we get left with this molecule, this heme, that we have to do something. And so in the macrophage, there are some enzymes that can turn heme into biliverdin. And I think in Spanish and French, verdin is verdine, I don't know, it's green. So part of what it looks like initially is a green colored molecule. And then it's converted again to bilirubin. And bilirubin can now circulate in our plasma. So right now, pull out some blood, centrifuge it. I should find a little bit of bilirubin in there. And bilirubin goes to the liver, and majority of the bilirubin is pulled out and becomes part of your bile, which will then be stored in the gallbladder and be shot into your digestive tract, especially when you eat fat foods, fat-rich foods. A little bit of your bilirubin can be pulled into your urine, and it's part of why urine takes on a yellowish color. The rest of the bilirubin, when it's shot into your gut, it's going to help you emulsify fats and help fats be absorbed. Not all of the bilirubin, hopefully, will be absorbed back. So that bilirubin will eventually end up in the large intestines, and bacteria in there are going to then use it as a molecule that they make energy out of, and it'll become stereobilins, urobilins, and it'll end up making your poop turn different colors, right? Brownish and yellowish colors. Okay, so that's the life cycle of a red blood cell. And again, we take out about 1% of our red blood cells. Somehow macrophages know what an old red blood cell looks like, and we bring in young red blood cells. And there is something we'll learn again in respiratory. Young red blood cells have a little bit more of pyruvate is in low supply and high supply of glucose and the molecules in glycolysis right before where you make energy. So some 1,3-biphosphoglycerate. So maybe macrophages can figure out that a young red blood cell has a lot of 2,3 and 1,3 BPG. Again, that's a molecule that's kind of in the upper part of glycolysis. And an old red blood cell doesn't have any more BPG. It has almost all pyruvate. Again, that's inside the cell. I don't know how the macrophage knows stuff inside the cell. So maybe there's something related to the antigens, the A protein, the B protein, or the RH protein, or other antigens that get put on the red blood cell that we then, the macrophage can figure out you're old or you're young. Okay? All right. Red blood cells get made again in the bone marrow. That hematopoietic stem cell again gets signals through a bunch of different proteins to be able to go through cell division and cell cycle. One of the key ones that lead it to the red blood cell pathway is EPO, and that stands for erythropoietin. Again, we talked about this last semester in our hormones of the kidney. And we'll go through a little bit more. Hopefully I can hit that in lab this week, or you'll hear more about how EPO regulation works with like hypoxia and oxygenation of the cells. But it's made in the kidney, and the kidney is really, really unique in that it only has one artery in. And so the blood that the kidney gets is going to be all the red blood cells that it gets access to. And if the kidney sees that you are not delivering enough oxygen in that million red blood cells it gets per minute, its only course of action is to tell your body to make more red blood cells. And that's why it's the main place EPO comes from. Okay. There are other molecules that work with EPO. That's why I can't take stem cells out of the body, put them in a petri dish, and grow red blood cells for me to then give people in the hospital. That's why we still need blood donations, because we haven't figured out the perfect cocktail of EPO and what other factors make all of my stem cells make red blood cells. If and when we figure that out, then we won't need people to donate blood anymore, because we can grow it in the lab, but we're not there yet. All right. And again, going back to these antigens and what red blood cells display on their membrane, they are a cell, and just like every cell of your body, they're going to put proteins on their membrane based on the genetics that's in your DNA. And so A blood type means you are going to have a gene that says you make the A antigen, and the A antigen is going to show up on your red blood cell membrane. And then if I know that you have the A antigen, if I take your blood and I put it in a well and I put on purpose A anti-A antigen antibody on it, I should then see the antibody and the antigen meet, and I would then know cells have A antigen on them, so you have the A gene in your body. All right, so this is what blood typing is really telling me. It's telling me a little bit about, one, what genes are in my DNA. Two, it's telling me what proteins are on my red blood cell. And because not every human has the exact same DNA, I get to start to see as a general population what most of the people in my population might have for genes. It used to be O was the most popular blood type. I think now with all the intermarrying and more world travel, A and B and A are beginning to rise up as more people begin to, again, marry people with different genetics and produce offspring of higher potential of A and B blood types. All right, the RH factor as the little, it used to be a big deal if people would have one kid and they never could carry a second kid to term. And that's a different gene and a different location, but it still produces an antibody or an antigen on your red blood cells. And if you're positive for the RH factor, you'll write someone's blood type as A, so they have the A antigen, and they have the RH factor, so that's an A positive. If you do not have the RH protein on your membrane, you would then be just A negative. Okay, so normally when you do blood types, you need to give the letter of the gene they have, and the lack of that gene would be an O or a void. so O blood type, and then you give them a positive or a negative, again, to indicate on a different gene, on a different location in the DNA, do they have this other protein on their plasma membrane or not? All right, so last point, do we have any questions? In lab, I will ask you to learn these abbreviations and definitions, and you don't have to worry about this over here, but one of the things, these are some good review questions as you go through the first part of chapter 19 is, do you understand how blood typing works? Do you understand where red blood cells come from? What influences their numbers, their production? Remember, boys, testosterone is going to make you have more than females. The structure and the contents and why that's important, the typical lifespan, and the process of breaking down and where it occurs. Do you know what I can't even read that because my eyes are terrible and I haven't got my new glasses. What's MCV and MCHC? You need to know what these abbreviations are and mean. In lab manual, it has you do some calculations. We don't do math on lab exams, so no, you will not have to calculate them, but you need to know what these abbreviations mean and what they represent, just like you need to know what serum versus plasma means. Okay? Any questions? Okay, hopefully, again, I won't take as long. I'd like to keep my lectures at 50 minutes or less, but thank you guys for coming. Thank you especially to all my 212 and 696 people. Know that the links I sent you today should be the link for every Monday, and the link you have now listed in that announcement for Wednesday and Friday should be the working links for the rest of the semester for Wednesdays and Fridays. PowerPoints are all in the weekly materials. So let me see if I can share my screen. If you go to the Blackboard, I don't know if I can do share application or screen. Okay. I'm trying to do this. Okay, so I don't know. All right, if you go to your Blackboard site, I don't think I can share it and get to it. Under the week, there should be cardiovascular unit 1 content. There should be PowerPoint slides. The slides are going to be the original blood slides, so they are going to have more of the slides listed that I combined into one slide. I made 4 things into one slide. So you have those available, and when you go to my YouTube channel, the lectures are going to be with those slides, so you'll be able to follow along with them that way. Any other questions? Okay, when in doubt, read the book, read the chapter, and my office hour, again, you should be able to email me if you want to try to do a Zoom or a Microsoft Teams meeting. Or we can just respond by email. But I will try to do half hour before and after labs, make myself available. And then I will try to maybe even have a Microsoft Teams thing where you could come and try to live chat with me on Thursday mornings. I'm trying to set it up to maybe to do between 8:30 and 9:30. It just all depends on how crazy the kids are come Wednesday when they start school. Okay, any other questions? We will have to label things. Label things as lab. Yes, you will be labeling. That's what the ADI labs are about. So if you have not gotten the ADI LT invite, you need to email me so I can make sure your STLCC email gets to you with that invite. Homework is the ADI labs every week. You don't have one week 1. They start week 2. For quizzes, again, there might be some things that you might have to label in the quiz. Chances are most of the time the quizzes are going to be more lecture-ish in nature of questions that you might see on lecture exams. And the ADI LT lab activities are going to be your practice for labeling activities. Okay. Hopefully those answers a lot of questions. You're still expected to memorize and know features, anatomical positions, landmarks, locations of organs, and that would be more on a lab exam, not necessarily a lecture exam. All right, I'm going to stop the recording.